And so these videos is on membrane lipids, so cell membrane itself. So let's just look over the cell and the organelles and structures within it. So here we have the nucleus where the DNA is stored. We have the rough endoplasmic reticulum around the nucleus. We have mitochondria, the smooth endoplasmic reticulum with the ribosomes. We have the Golgi apparatus, lysosome, and we are concentrating on this part, which are the membrane lipids. So if we zoom into the structure here, a typical lipid membrane, cell membrane, what role does it have in the cell? So here we have the typical cell membrane. We have the extracellular fluid, the outside of the cell, and we have the intracellular fluid, the inside, the cytoplasm. Now a typical cell membrane is about 6 to 10 nanometers thick. A typical cell membrane also consists of proteins and lipids. So here's a protein and here's a lipid. And also actually carbohydrate chains or monosaccharides are linked to the proteins or fats itself. And they have a special role. Now the protein structures, they actually have a special function or role within the cell, such as being a receptor, for example. So let's look more closely on the functions and the roles cell membranes have. And this can be on the outside or the organelle's membrane itself. Remember, there's a wide variety. So, 5 points. Number 1, cell membrane defines cellular boundaries. So, the statement is obvious, it just defines the boundaries. So it protects the cell, you can say. 2, it provides intracellular boundaries. So organelles have boundaries, such as the nucleus with the DNA, or mitochondria. Now 3, it can organize complex reactions, such as the receptors, ion channels, for example. 4, it can regulate flow of information. So it can send out signals as cytokines and communicate with other cells, receptors, etc. And lastly, 5, they have dynamic structures, and we'll talk about these later on. But for now, let's zoom into the cell membrane. The cell membrane, the plasma membrane, is a bilayer, which means it has 2 layers. And it's a planar sheet, so it's continuous, like this lipid membrane. And we'll talk more about the lipid bilayer later on, and the evidence that proves that the membrane is a bilayer. But for now, let's look at one of these lipid molecules that form the membrane lipid structure. And you can say that the shape of it is cylindrical, surface area, and it's also amphipathic. What does this mean? It means that one side of the lipid loves water, and the other side hates water. And this structure enables it to form a bilayer spontaneously, if you know what I mean. And the membrane are actually self-forming itself. So what does this mean? Let's look at an example, at a normal lipid structure. A normal lipid structure contains a carboxyl end and a long fatty acid chain. The carboxyl end is hydrophilic, you can say. It loves water. And the chain, hydrocarbon chain, or the alkyl chain, hates water, hydrophobic. So what happens if you put this in a solution of water where water molecules reside? Well, the water molecules will start surrounding the hydrocarbon chain and will not form bonds with it because it's hydrophobic. Right? Okay, so essentially water molecules will form cages around the hydrophobic alkyl chain of the fatty acid, because it can't interact. So it will just form cages. So for example, if we have two lipids in solution, the water molecules will surround it, will cage it like so, and will not form bonds with it because the alkyl group is hydrophobic. And this means that the lipid molecule, the hydrocarbon chain, will force water molecules to become highly ordered. So it uses energy. So what happens if we combine these two lipids together to form a cluster of lipid molecules? Well, this means that the water fewer water molecules are ordered, and therefore entropy increases, because the water will now, only part of the water will surround the lipid structure and will not be in the middle, because the lipid will prevent this. So now, if we have many clusters of lipid molecules, like so, and we basically connect them up all together, it will form this sort of shape, where all the hydrophobic regions, regions that hate water, will be inside, and all the hydrophilic regions will be outside, forming what's called a micelle. And the hydrophobic groups are together, and the water molecules are minimized. So entropy is increased. So looking at one of these structures, this is a cone-looking structure, not like the previous cylindrical structure, if you remember. And the cylindrical structure is from the bilayer, the membrane bilayer. And the difference is that the cone consists only of one tail, right? But the bilayer consists typically of two tails, and it's cylindrical. And this is what forms the bilayer. And if we put all the bilayers together, it will form and if we form a circle out of it, it can form a vesicle. So all the hydrophilic regions aiming towards the water region, and all the hydrophobic regions in the inside. And this inner circle is aqueous solution, so it consists of water. And so perhaps organelles can be in here, forming what's called a cell. So going back now to the cell diagram, we'll zoom into the plasma membrane here. Now the plasma membrane, we are looking at what's called the fluid mosaic model. Now what is a fluid mosaic model? Now it's a basic model for the structures within the plasma membrane. And so now let us look at some typical structures and features of a typical plasma membrane of a cell. Here's the cytosol and extracellular fluid. Now the first important structure I'd like to talk about are these red things, which are sterols. And it's important for the membrane, such as cholesterol. And we'll look at why this is important later on. Of course, this is a bilayer, as mentioned. An important component of the membrane also are the proteins, such as the peripheral protein, this structure here, which faces into the cytosol. And there's also another protein, peripheral protein, but this one is lipid linked. As you can see, it's connected with the lipid. Another protein that goes through the membrane is what's called the integral protein. And this integral protein is a single transmembrane helix, which means that it's a single helix here. And the helix is hydrophobic, and the outer parts are hydrophilic. We also have glycoproteins and glycolipids, which are carbohydrates attached to proteins and lipids. So that's all we're going to talk about on the fluid mosaic model, and we'll look at each of these structures more closely later on throughout this series. But for now, let's look at the structures within the inner leaflet of the cell membrane. So in the cytoplasm, the inner leaflet of the membrane. And we're looking at a specific cell called an erythrocyte, which is a red blood cell. And erythrocytes contain ion channels, which control the movement of ions in and out of the cell, for example. And here's an ion channel. So this cell, let's just say, we are bending it, and we're looking at the inner leaflet of the membrane, so the inside, and I'm trying to depict it here. So the inner leaflet of the membrane contains structures such as actin, which is a protein and helps in maintaining the cell shape. Also, the inner leaflet and the cytoplasm contains things called cytoskeletons, which restrict motion. And a particular cytoskeleton in erythrocytes are called spectrins, which help in membrane flexibility. And that concludes part 1 of the cell membrane video series. Next video, we'll look at what evidence there is to support the fluid mosaic model, as well as other features of the cell membrane. So please watch part 2. Thank you for watching. Please like, comment, and subscribe. Now this is part 2 of the cell membrane series, and we're looking at the evidence of the fluid mosaic model. So what evidence is there to support this? Well, if we have a cell and we freeze a cell, for example, and then we fracture it with a knife, you can say, we can be able to split the membrane, because part of the membrane will go on one side of the eyes and the other will go on the other side, like so. And so using this, if we zoom into this section, you can see here is one layer of the lipid membrane, and here's the other layer. And so you can see that the lipid membrane is peeling, and you can do this by using scanning by electron microscope. So if we zoom into the cell membrane again here, the plasma membrane specifically, we can appreciate the sense of scale of the membrane, this plasma membrane specifically. And so, as mentioned, the plasma membrane is about 6 to 10 nanometers. A DNA in our body is 2 nanometers, so not that much difference. And a ribosome in the cytoplasm is about 25 nanometers, and these transcribe proteins, remember. So as you can see, the plasma membrane is pretty small. So if we look at one of these lipid structures again, just going over it again, just recapping, it's amphipathic, which means that one side loves water, the head group, and faces extracellularly, and the other side, the fatty acids here, hate water. So here, the fatty acids, they're hydrophobic, they don't like water. The head group is hydrophilic, it loves water. And we are concentrating on the head group because the head group can change. There can be different chemical groups attached to the head group, which changes its charge, structure, etc. So essentially, the membrane lipid, there are 3 types, or 3 categories, not 3 types. And the main ones, the head groups can be a phosphate to it or a carbohydrate to it. So one category, the membrane lipid, can be a phospholipid with a phosphate, a glycolipid with a carbohydrate, or it can be a sterol, which is, yeah. And sterols, essentially, which we will talk about first, is just cholesterol or ergosterol. And there are many others, and we'll talk about these later on. But for now, let's concentrate on phospholipids and glycolipids. Now, phospholipids contain phosphate and can be divided again to 2 categories, to 2 parts. There can be glycerol phospholipids, and there can be sphingolipids. And they both contain phosphate on the head group. Remember, they contain phosphate on the head group. So glycerol phospholipids essentially contain a glycerol backbone with 2 fatty acid tails and a head group, a phosphate head group with X being any chemical group attached to it. A sphingolipid has, instead of a glycerol, a sphingosine backbone, 1 fatty acid tail, a phosphate, and an X. In this case, it's a C, which stands for choline. So it's a sphingolipid choline. Now, a glycolipid is similar. It can be divided into 2 parts again. There can be sphingolipids as well, or galactolipids, sulfate particularly. Sphingolipids has the sphingosine backbone and 1 fatty acid tail. And instead of a phosphate, it has carbohydrates, a mono or oligosaccharides. A galactolipid contains a glycerol backbone, same as a glycerol phospholipid. Glycerol backbone with 2 fatty acid tails, and again, a carbohydrate chain. And in this case, particularly a sulfate head group. And it should be noted that there are much more phospholipids in the cell than there are glycolipids. And that's why you might hear cell membrane being referred to as a phospholipid bilayer, because there's more phospholipids, and we'll soon see why. But essentially, these are not the actual structures of the cell membrane. The head group should be facing one way, and the fatty acid tail should be facing the other. So if we look back at one lipid membrane structure again, we know that there's a hydrophilic group, the head group, and a hydrophobic group, the tail, the hydrocarbon tails. Right? So if we look back at the glycerol phospholipid, we have the glycerol backbone, the hydrocarbon, the fatty acid tails, facing down, and the head group, the phosphate, with the X facing up, facing the water. So it's similar in structure. Same with the sphingolipids. We have the sphingosine backbone facing down, essentially, with the fatty acid with 1 fatty acid tail, and the phosphate with the choline group in this case facing up, facing the water, facing the extracellular fluid or the cytoplasm. So hydrophilic and hydrophobic. This is same with the glycolipids, containing the carbohydrates. the sphingosine backbone, and the fatty acid tail facing down, and the carbohydrate, either monosaccharide or oligosaccharide, facing up, facing the water. Galactolipids as well, the glycerol backbone with the two fatty acid tails facing down, and the carbohydrate chain facing up, facing the water. Now let us look at the different types of phospholipids and glycolipids there are found in the cell, the main ones. So as mentioned, the phospholipids can be divided into two classes, the phosphoglycerides, or the glycerol phospholipids, and the sphingolipids. The glycolipids can be divided into sphingolipids, and we won't talk about the galactolipids here. And also there are sterols, and we mentioned particularly cholesterol and ergosterol, and we'll talk about cholesterol later on part 3, I think, or part 4. So phosphoglycerides, there are many types. It begins with phosphatidyl. So there's phosphatidylcholine, there's phosphatidylethanolamine, there's phosphatidylserine, there's phosphatidyl'inositol, and there's phosphatidylglycerol. That's one class. And the other one, there's cardiolipin. And we'll look at each one of these closer later on. The sphingolipids, there are two main ones. There's the sphingomyelin and sphingocholine. For the glycolipids, the sphingolipids, there are cerebrolipids and there's gangliosides, or cerebrosides and gangliosides. But we will be mainly concentrating on the phospholipids because there's more plentiful phospholipids in the cell membrane, I would say. So we'll look mainly at the phosphatidyl group and also the sphingomyelin. Now, talking about something different, this is a neuron, as you know, and a neuron contains myelin sheath. And now we will be looking at the different components of a cell, of the different membranes within a cell, including the organelles. So what we're looking at specifically are the components as in proteins, which are designated as black box, and lipids, which are designated as a red box. So the components will be in percentage from 0 to 100%. First is the myelin, which is essentially the myelin sheath wrapping around the exon. And it's composed predominantly of lipids, about 80% lipids, and the rest proteins, 20% proteins. Next we have the Golgi apparatus membrane. We're looking at the membranes, remember? And the Golgi apparatus membranes is composed about 55% lipids and 45% proteins. And remember, these are figures from a basic cell, not only a neuron, except the myelin, which is obviously a neuron. Plasma cell is composed about 50% proteins and 50% lipids. Now the outer mitochondrial membrane, because remember, the mitochondria has two membranes, the inner and outer, but the outer mitochondrial membrane is actually about 50% lipids and 50% proteins as well, similar to the plasma membrane. And then we have the rough endoplasmic reticulum membrane, which is a bit less, maybe about 45% lipids and about 55% proteins. And then we have the nucleus, which is predominantly proteins, about 60 to 65%, and the rest lipids. And the inner mitochondrial membrane especially is composed predominantly of proteins, about 80% proteins and about 20% lipids. So it's very different to the myelin. So from this diagram you can appreciate how the organelles within a cell are composed of varying amounts of proteins and lipids. So that concludes this video. Our next video will look at the differences between the membranes in the organelles and around the cell. Thank you for watching. Please like, comment, and subscribe. Thanks. And so we're continuing on with the cell membrane videos. We're looking back at saturated fatty acids and unsaturated fatty acids. So what are the differences? So saturated fatty acids, carboxyl end with a hydrocarbon tail. So essentially it would look like this. Unsaturated fatty acid has a double bond in the carbon chain and so forms a kink. So what does this mean? Well, it means two things. So the first one, the more longer the fatty tail is without a double bond, it means that the higher the melting point will be. So with if it's saturated fatty acids, the longer it is, the higher the melting point. Number 2, however, the more double bonds in the fatty acid tail, the lower the melting point will be. And so if you think about it, it means that the unsaturated fatty acid tail can be degraded quicker. And so sometimes scientists or people think that unsaturated fat is better. And you can be the judge of that. Okay, now let us put these unsaturated fatty acids and saturated fatty acids into context by putting it on the lipid membrane bilayer. So as you can see, we have some saturated fatty acid tails and some unsaturated, which forms kinks. Here is an integral protein which goes across the membrane, peripheral protein. In the intracellular layer, we have also carbohydrates coming off the protein and the fats. Let's step away from the saturated and unsaturated fatty acids and now look at the structures of the lipid membrane bilayer. So one important thing is that glycolipids and glycoproteins always face out, facing the extracellular fluid. 2, the peripheral proteins always face the cytoplasm usually. And 3, the lipid distribution is asymmetrical. So there's this different distribution of the different types of lipids on the first layer, the top layer, and the bottom layer. Let's go back to the different types of the lipid membranes. We have the phospholipids, remember, and the glycolipids. Glycolipids are cerebrosides and gangliosides. And then we have also the sterols, composed of cholesterol, which we're about to look into later. The phospholipids are composed of the sphingolipids and the phosphoglycerides. The phosphoglycerides, we have phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, phosphatidylglycerol, and cardiolipin. For the sphingolipids, we have sphingomyelin. So these are the different types of lipid membranes, right? So going back here again, number 3, there's lipid distribution. The lipid distribution is asymmetrical. So to portray this example, I'll draw a graph here. So we have a scale 100% on the top, which composes the outer membrane, and 100% to the bottom, which composes the inner membrane. So, for example, this is a red blood cell plasma membrane. So let's look at the different components that are in the red blood cell plasma membrane, like the phosphoglycerides, and how much of it is composed on the outer membrane and the inner membrane in respect with the inner membrane. So for example, here we have phosphatidylethanolamine, and as you can see, there's higher concentrations of phosphatidylethanolamine in the inner membrane. Phosphatidylinositol, which is a second messenger, has also higher concentrations on the inner membrane. They're on the inner layer. Phosphatidylserine, which is very similar to phosphatidylinositol, there's higher concentrations on the inner leaflet. Phosphatidylcholine, on the other hand, there's higher concentration of phosphatidylcholine on the outer membrane leaflet, as well as sphingomyelin, which is highly concentrated on the outer membrane. So as you can see, the distribution of the different types of lipids vary in the red blood cell plasma membrane. Now this is going to be a short video on membrane dynamics and mobility of membrane components. So how components within the cell membrane move around. We talked about cholesterol before moving around. Now we can talk about the lipids moving around. So membrane lipid dynamics. So here I'll draw the lipid bilayer again. We have the extracellular fluid here, outside, and the intracellular fluid, the inside. So there are 2 mechanisms of membrane lipid dynamics. There's uncatalyzed and there's catalyzed. In uncatalyzed, we have what's called the trans-bilayer diffusion, also called flip-flop, which is a funny name. And this process is actually very slow and takes days. So this is essentially when a lipid from one layer moves to another layer. The second type of uncatalyzed movement dynamics is what's called the lateral diffusion. And this process is very fast because the lipid structure essentially moves within the same plane on the same layer, and that is why it is fast. Now let's look at the catalyzed membrane lipid dynamics, which uses proteins, enzymes, to move lipids from one layer to the other, which will obviously lower the activation energy. So this process will be faster than the trans-bilayer diffusion process, which is very slow. So for example, if we have the extracellular fluid here, the enzyme flippase, which is the process also called flippase, requires ATP and essentially moves a lipid on the outer leaflet to the inner leaflet, so outer to inner leaflet. And as mentioned, it requires ATP. The second requires floppase, the enzyme floppase, the protein, the process. And this is the reverse of flippase. This is when the lipid in the inner leaflet moves to the outer leaflet with the help of this protein. And this process also requires ATP. The last is called scramblase. And this is a very interesting name, but essentially there's an exchange. A lipid on the inner leaflet moves to the outer leaflet, and at the same time, a lipid on the outer leaflet moves to the inner leaflet. Now, there are other types of movements within the membrane structure. If we have a look on the top of a membrane, for example, so like a bird's eye view, and we zoom into the structure, the lipid membrane will look like this from the top view. So here's a top view of the membrane. Now there are two things I want to point out here. One is that the lipid can move around on the same plane. And this is sort of like lateral diffusion from the uncatalyzed version, but essentially it can move anywhere within the same layer, around the same plane. And two, the lipid itself can rotate, obviously, because it is in constant motion. And finally, I would like to mention that the fatty acid chain can actually flex, can move around, because there's a sort of a fluid environment within the bilayer. Interesting. Now let's talk about how membrane fluidity itself is influenced. Now, what influences membrane fluidity, most importantly, the two, is temperature and also the fatty acid composition. What I mean by this is the fatty acid length, for example, or if they contain unsaturated double bonds. So to show you an example, I would like to draw you two membrane bilayers. So the interior consists of some form of fluid and it's in constant motion. So if you can imagine that the fatty acid chain is just moving within the bilayer. And this is also enhanced with increased temperature. And also an important feature is the fatty acid composition, because if you have unsaturated fatty acids, it's more flexible because it melts quicker than saturated fatty acids. Just think about it. Now, if we, for example, decrease the temperature of within the membrane, this will cause the whole structure to stiffen, you can say. And so it's not as fluid. And this state is called a paracrystalline state. And it changes the membrane, cell membrane length in some way to become longer because it's stiff. Now, other things that influence membrane fluidity is the phospholipid classes, such as if there's heaps of phosphatidylcholines, phosphatidylserines, or phosphatidylinositols. Sterols can also influence this, and lipid-protein ratio, if there's a lot of proteins or if there's a lot of lipids within the cell membrane. Now let's talk more about the membrane fluidity and what it's influenced by, and look at other factors, such as environmental conditions. And as mentioned before, temperature plays a vital role in the environment, and we also have the ionic strength within the area, whether there's a lot of ions around, the acidity, if there's solvents present such as ethanol, and if there's free radical and oxidants, which can potentially damage the membrane. A second important influencer is the protectant molecules, and this will help obviously protect the membrane, and also the cell's energy status and metabolism. So whether the cell needs energy, and so this influences the permeability, etc. And another important thing, which is also to do with basically everything that I just mentioned, is membrane proteins. Because membrane proteins are enzymes, transporters, and also essentially increases permeability of certain ions and basically helps maintain the membrane structure. It has a vital role in protecting the membrane structure as well. So membrane proteins. Now the fluidity must be optimal for the function of membrane proteins. And we'll look into membrane proteins in the next video in more detail. But let's just look at a quick overview. What are membrane proteins? Well, they can be transporters, they can be receptors, energy transducers, they can be enzymes, and obviously they can influence permeability. So this video was talking about membrane dynamics and lipid membrane dynamics. Next video we'll look at the membrane proteins in more greater detail. Please like, comment, and subscribe. Shankyo.